Ew. Okay, I think I've sort of figured out a better light situation where it just gets a... No, wrong one. Yeah. Is that any better? So it's not quite as dark? Okay, great. Okay, well, thank you everyone for coming to the last session on genetics. And to those of you who this is your first one, hopefully it'll make some sense. I'll do my best. And as people know that have been here before, just ask me at any time to slow down, because I can talk fast, or repeat anything, or ask any question that obviously you have. Anyone who's new, there are microphones in front of you so that if you push once on the little button and it lights up red, then on the video taping or recording of this, I just talked to the tech guy and I said, I want this video taped. And he said, you just dated yourself. It's not video taped. There is no tape. I'm like, once you push that button, it will end up in the recording so people can hear the question and then they can hear my answers. So if you do that, that would be helpful. So I'm going to finish up. Yes.
I was just wondering, we didn't quite get to Alzheimer's last week?
Yes, so I was just going to say, thank you. Yeah, so I'm going to finish up what I didn't get to last week.
Great.
So I have a tendency not to finish what I did the week before, and so I'm going to finish what I did, and I made this week nice and reasonable and short so that we'll get to everything. And I wanted to leave some time in case there were further questions or anything that someone wanted to ask a little bit more about. And to leave it open for this option that I thought might be interesting, that we talked last time about the 23andMe that you can order, that you can send in and get your genetic information. That one I've sort of largely warned you about, that you know, if an insurance company then asks you, do you know anything about your health now, you will know way too much about your health, and it would be difficult for insurance companies to insure you. But I did it because I'm foolish, and I was thinking it might be interesting to show you what it might look like and what some of the traits are that I have information about and some of the mutations that I carry, just so that you can see what it might look like if it were of any interest to you if we have some time. So I tried to make this nice and short for that reason, so we have more time for that sort of thing. So yes, I'll pick up where we left off last time and start with the Alzheimer's. So just to remind you, we're in week 4. So now, once I speak about Alzheimer's and a little bit about, I added some more information in based on questions I had last week. So about stem cell treatment for these things, and then also we were talking about obesity and some specific genes for obesity, and if there is a genetic component of obesity. So I have some information and a little video on that too. And then we'll move into this idea of how medicine can work with your genetics to improve your care. So where we're currently right now with the genetic information and how that is proposing and will eventually help with medical care so that things are more tailored to you. So that's what I'll talk about near the end. So again, just to go over, continuation from last week, stem cell treatments for both Alzheimer's and Parkinson's disease, and I'll talk a little bit about what they both are, of course. Genes associated with obesity and overeating, because we certainly know those exist. And then personalized medicine, what that means, what that looks like right now, and how the Human Genome Project is related to that. And then pharmacogenetics, which is looking into tailoring your the pharmacology, so the drugs that you would take based on your genetics. So there's a lot of research into that right now, knowing that certain drugs don't affect people the same way. And so using our genetics to make sure that we get the proper treatments. So just to remind you, and I don't think we even really talked at all about Alzheimer's disease, not completely. So Alzheimer's, as some of you may know, so it is, what happens in Alzheimer's is a development, I think we talked about this maybe 2 weeks ago, of these plaques, these amyloid plaques, and these tangles of these fibrils. So it looks something like this, where everything gets all tangled up. So these proteins get tangled up and form tangles and plaques in the brain. And then because of that, there are loss of connections between the neurons in the brain. And the neurons are just brain cells or nerve cells, and they have to communicate with each other in order for us to do everything that we do. So speaking, breathing, walking, everything, those connections need to be intact. And so with the loss of those connections, some of the symptoms arise. And is also death of nerve cells too. So some of the nerve cells and neurons themselves die, and then they can't function, of course. And we used to think zero regeneration of nerve cells. We know that's not completely true now. There's some growth of new nerve cells, but mostly not. Once you lose nervous tissue and brain tissue, it's gone. And so that death from Alzheimer's means that you will lose some function. So both early and late onset Alzheimer's have genetic components. And in the next slide, I'll talk about what the difference between those are, but they're different diseases, essentially. Their outcome is the same, but the genetics, one is a very early onset and one is a later onset, one that we're more used to thinking about and knowing about. But there are a few risk factors for genes that are implicated. So I mentioned this when we talked about Craig Ventnor, who was the scientist who had his own genome sequenced as part of the Human Genome Project. He found out inadvertently that he has this ApoE4 mutation. And so he didn't know he was going to find that out. And now he lives with the knowledge that he has a mutation that is a risk for an association with an increased risk for Alzheimer's. So it's called apolipoprotein. So lipo means fat, protein means protein, so it's a fat protein. It's version E and then allele E4. So remember we talked about alleles way back 3 weeks ago or 4 weeks ago now, that alleles are just different versions of a gene. So there are alleles for brown eyes, blue eyes, green eyes. This is just an allele. This E is an allele, this E4 of ApoE, apolipoprotein E, and it's associated with Alzheimer's. So it's the strongest influence that we know, and it's thought to contribute to about 20 to 25% of Alzheimer's cases. So individuals with ApoE4 have an increased risk. And interestingly, this is on chromosome 21. And so remember we talked about what's interesting about chromosome 21, or what's this syndrome that's associated with chromosome 21? Does anyone know? Down syndrome. Yeah, so individuals with Down syndrome have 3 chromosome 21s. And interestingly, they are at increased risk for getting Alzheimer's. So it seems to make some sense that if you have 3 copies of even, they tend to have E4, and then they have 3 copies of it. So they're more prone to getting Alzheimer's than the average person in the population. Chromosome 21 is very interesting for things like that. There's lots of other physical things that are associated with Down syndrome because of important genes on chromosome 21. So early onset Alzheimer's, or familial Alzheimer's disease, FAD, it occurs at ages 30, as early as 30 to 60, and that's less than 5% of Alzheimer's patients. So it's very much less common, but it does happen. So individuals at 30 years old up to 60 could get Alzheimer's. The child of a carrier patient has a 50-50 chance of inheriting that mutation. So this goes back to 2 weeks ago when we were talking about the genetics of how things are inherited. So the predisposition, a carrier parent, do you remember what that means if someone's a carrier? Yeah, so they just carry the mutated gene, but they don't actually have the disorder themselves, or they might get it at some point, but they don't present it yet. That's what a carrier means, is they carry the mutation. Then there's a 50-50 chance that their child will inherit it from them. And so that's because they have 2 chromosomes, one of which will go to their child. So 50-50 chance that the child will get it. It's caused by a single gene mutation the early onset is on chromosome 21, 14, and 1. So those are the important chromosomes in early onset Alzheimer's. On chromosome 21, what happens when that's mutated is you get this abnormal amyloid precursor protein, or APP. And that's when we go back a second and just take a look at it's these amyloid plaques that are causing some of the problems in Alzheimer's. So this mutation on chromosome 21 is an abnormal early protein version of that amyloid. So it's abnormal, and then the protein becomes abnormal and forms these plaques in the brain. So that mutation on chromosome 21 causes that to happen. On chromosome 14, it's an abnormal presenilin 1, which is a protein. And then on chromosome 1, it's an abnormal presenilin 2, which is a protein. So all of these mutations, as all mutations are mutations in genes that result in an abnormal protein that doesn't function properly. And in this case, it causes tangles and plaques in the brain and death of some of the tissues in the brain. So the mutations break down this APP, and the function of that protein is not fully understood, but that's what causes these amyloid plaques, and they're harmful. So they destroy tissue in the brain. So there's a large genetic component to that we know. Any questions about the early Alzheimer's before we move on? Yes.
Jane, can you explain the evolution or origin? Why does it happen? I mean, why does it cause the premature death of a lot of seniors? How did it evolve?
Who knows? So evolution is all these mutations and how things evolve are not known. So it's just a mutation happens, and then it causes a problem in a gene and causes a problem in a protein, and it happens. So it's just like, you could ask that question of any disease, like how did any mutation happen? It's just random chance. So all mutations are random chance. It could be that you were exposed to radiation. It could be you were exposed to some food that damaged the DNA, or as we've talked about over the weeks, the system that replicates DNA just makes mistakes. And so it's just random. And what happens is that at some point in our history, a human had this mutation and then had children that inherited the mutation, and now we've inherited it into present day. And who knows when it started, but it was just all mutations are just random. Yeah. And there are lots and lots of mutations that happen that then don't ever make it into the next generations because those individuals can't reproduce or don't live long enough to reproduce. It's just in this case, it's a late onset disease, so people pass it on. So most people that have Alzheimer's let's say I suddenly got Alzheimer's when I was 50 and I had early onset Alzheimer's. I wouldn't necessarily know. It doesn't mean that anybody in my family has to have either had it. It could be a new mutation, or they might have late onset Alzheimer's and they just don't know it yet. So they had me and they had all these children, and then suddenly Alzheimer's shows up in the family. And so, this gets into all sorts of things about, we continue to allow the mutations to move through generations, largely because there's nothing we can do about it unless we stop people from having children who have diseases, but we all carry something. So.
Is there any medical test for that?
Sorry?
Medical test.
Yes. Yep. So those mutations that I was talking about, the ApoE in particular, you can test for that. And so that's why I was saying that Craig Ventnor, he found out when he had his genome sequence that he carries this mutation. He knows he's at an increased risk. And I can show you, I can't remember. This is how important it is to me. I can't remember if I had the ApoE4 or not. I don't think so. But this is the one I think I told you in that 23andMe where there's a locked, closed section where they warn you, are you sure you want these results? They contain very sensitive results. It's about Alzheimer's and breast cancer. And there was one more that I can't ever remember. It's not Huntington's, but just because I carry this mutation, if I do, doesn't mean I will get it. It could be that I will never get it, and it could be that I live to 75 and I was going to get it at 77, but I passed away before that happened. So it's not critical, but we do know a little bit about it. Okay, so then late onset Alzheimer's is what we're most used to thinking about and hearing about. Occurs after the age of 60, and so it's much more common. It's a combination, as the other one is too, but of genes, environment, and lifestyle. So all things play a role. So the ApoE gene on chromosome 19 is a presumed risk factor. So, it's on 19. Sorry, I made a mistake. It's the amyloid that's on 21. So ApoE has several forms. Those are the alleles that I was talking about. There's E2, E3, and E4. And it's E4 that provides this increased risk for Alzheimer's. And it's also associated with earlier onset. So you can have 0, 1, or 2 of these ApoE4 alleles. And the more you have, the more increased risk you're at. So if both of your parents were carriers and you end up with 2 versions of ApoE4, you're much more likely, because there's larger genetic component now leading to you probably getting Alzheimer's. And so if you have Alzheimer's in your family, you can go and get genetic testing at a geneticist and a genetic counselor to find out your status of whether you have this mutation or not. And then late onset Huntington's. So we talked about Huntington's last time. That's okay. And I just wanted to talk a little bit about the details of Huntington's. So normally our neurons do not replicate. But in Huntington's disease, it's characterized by an accumulation of this Huntington protein because of a mutation in that gene. We talked about that last time. It builds up. It's abnormal and it just builds up in the brain. And then the overexpression of that protein leads to neurodegeneration. So you get enlarged ventricles and atrophy of the basal ganglia, which are responsible for motor movements. So if you lose those parts in your brain, that's why you get it's called Huntington's chorea, which is because it's an abnormal gait, an abnormal movement. And so the ventricles are this is a normal brain on this side, with the ventricles being these black parts in the middle here. So they're just sinuses or spaces in our brain, and they're enlarged in someone with Huntington's. And then atrophy just means degeneration or a shrinking of the basal ganglia and the nerve tissue in that area. And because our neurons don't normally replicate, once it's lost, it's lost. And so Huntington's is a progressive disease. It doesn't reverse. It doesn't get better. It progresses. So I'm going to show you a little video about stem cells, because I was asked last time about, isn't there research into some treatment for some of these diseases using stem cells? And it's really controversial because of where we need to get stem cells largely from, and also because we're talking about the brain and having to do major operation and put these cells into the brain, and how well does that work. So I just wanted to show a little video about that possibility and where we're at with that.
Yesterday, I taught 2 wilderness classes.
Oops.
See all the kids all staring at my arm, and so it reminds me to tell them that I have a disease that older people get that makes my arm shake, my hand shake. And this little girl yesterday, she came over and she held my hand and she tried to stop it from moving, and she says, "It does shake, doesn't it?" And I said, "Yes, it does." I first noticed my Parkinson's because of the tremor. And I read about the Parkinson's Institute and called and made an appointment. On the appointment slip, it says, "Why are you coming here?" And I put on there, "I want to know what I've got." And I saw Dr. Wong, and he diagnosed a definite Parkinson's.
Parkinson's disease is one of the major neurodegenerative diseases of aging, Alzheimer's being the other. Probably affects over 1 million people just in the United States alone. The classic symptoms most people will recognize: a shaking hand, slowness of gait, rigidity of the muscles, a stooped posture. It is a progressive disease, and it's incurable. We do have very good medications that can help on a day-to-day basis. The big problem in Parkinson's disease is that sooner or later we get side effects of therapy, and those can be worse than the disease itself. So ultimately, these therapies don't really help patients anymore. There's a big emphasis in our disease, in addition to medications, for exercise, dance, yoga. Anything that helps with movement and keeps patients moving seems to be of benefit in this disease, and we believe it's as important as the medications. There's a desperate need to find some way to intervene to stop the progression of the disease or reverse the progression of the disease. That's the major focus in the Parkinson's field right now. We can't get to the area of the brain in Parkinson's that's affected. It's deep in the brain. It's called the substantia nigra because it's not accessible. We would damage the patient irreparably. So we don't have a way to actually get real Parkinson's disease cells from humans. With the CIRM grant, we actually will have the funding to get cells from living Parkinson patients, skin cells say, drive them back to stem cells with new technologies, and then forward to those very same cells that die in Parkinson's. Once we've done this, we will have for the first time the opportunity to study disease mechanisms in a culture dish, a way we never could before, but I think even more importantly, to test for new drugs to treat the Parkinson's disease patients, and that could be revolutionary. One of the most important things, in my opinion, in terms of both medical research and patient care, is that they somehow communicate with each other. We can go from discoveries in the laboratory right up through clinical research and clinical trials to test new drugs without ever leaving the building. And it happens because it's a very seamless system here where all of us communicate and things can move very efficiently and easily right up from bench to bedside, if you will.
I'm not a technical person. I don't really understand all the research that's going on, but I volunteer for everything that comes along. I'm constantly in the nurse's office saying, you know, what's new, what can we do, and looking at their board up there. There is some urgency, as Michael J. Fox points out. There is some urgency. We're getting older, and we would like to see the cure in our lifetime.
Okay, so as you can see, it's not where we want it to be. It's really early stages, as that doctor was saying. They're hoping to be able to take those cells from a patient, turn them back to the stem cells, and make them back to the types of cells that we need them to be, which is the original nerve cells, but then we need a way to transplant those back into the brain. And as you could see from the diagram that he showed, it's deep in the brain. So you would damage the brain trying to get the rest of the brain trying to get to that area. So, and I think that's why he said the exercise is critically, and staying physically active is critically important, and then research for drugs to treat and change those cells and manage those cells in the dish is where the research is mostly headed. And so does everybody know who Michael J. Fox is that he was talking about? Yeah, so he's Canadian and an actor, and he has early onset Parkinson's, and he's had it for many years, and he's progressing quite quickly through in Parkinson's, and he's done amazing things for pushing this research forward and raising money and funds to do this. So obviously the early stages are there in research, but there has been some successful work done in mice, as lots of these things start off in mice, and there's been some success there. And the really good thing, as I said, it can be quite controversial because we have a tendency to think that the stem cells, as in early days, they came from terminated fetuses or something like that. And so people feel very nervous about, well, where do we get these stem cells from, and what sort of slippery slope are we going to end up on if that's what we do? But what he mentioned and what's happening now is that stem cells can be made from skin cells that are exposed to chemicals that turn them back to a stem cell. And so a stem cell is just a cell that has the potential to be any type of cell at all. So it's like a newborn cell. So it could be a skin cell, it could be a brain cell, it could be kidney, could be anything. And we're learning how to make those cells turn into anything we want them to based on chemicals that we provide it and nutrition that we provide it. So it's got a lot of hope for things like any of these neurodegenerative diseases, but also transplants. So there's a lot of work on making a kidney from stem cells and sort of imposing or on those cells to become a kidney that then you could transplant into somebody. We're far away from that happening, but that's the hope. And you may have seen things that have happened where we can make skin cells into the shape of an ear to replace an ear or things like that. Actually, they grow them on the back of rats sometimes. So I don't know if you've ever seen that, a rat with an ear, the outer part of the ear growing on the back. So that's what we've been able to do with a lot of these things, is stimulate normal, like you could take my skin cells, put them back to a stem cell, and then ask them to become something else, but with my DNA. So my body would never reject it, and it would be perfectly normal. So that's the hope. Yes?
I've nursed Parkinson's disease over years. Mm-hmm. And if my memory is right, when I worked nights, I don't remember the people with Parkinson's having tremors while they were sleeping.
Yeah.
Am I correct on that? Do they stop while they're asleep? And if they do you know why? What's happening in that brain?
Mm-hmm. So the question is, and the comment is that having nursed people with Parkinson's for many years, in the night, not remembering or noticing that they tremored when they were sleeping. And so is that true? And if it is true, why? And so I'm not 100% sure. I think my gut tells me that there are no tremors while you're sleeping. And I think it's just based on different stimulation from the brain to the muscles. But I'm not entirely sure. I'm not sure if anybody knows or happens to know anybody who has Parkinson's, or not sure. I would assume not. I would assume that there's some reprieve and that it's based on, it's an abnormal signal or an inability for that signal to get to the muscles. And so when your brain is, but there still probably are little tremors because your muscles are still working while you're sleeping. So it's interesting. It's a really good question. I'm not sure. Yeah. Mm-hmm. Umbilical cord, yeah, because those are blood cells and stem cells, and yeah, so that's a good place where people can get stem cells from. Yeah. Question over here? Yeah.
Was my question. I just wondered if you knew whether there are many people who are choosing to harvest the stem cells from the umbilical cord and have them stored. Do you know? Is that
Yep. So lots of people are. I just read an article a couple of days ago about how, as these things are, there's lots of controversy and concern about who has access to that blood and the DNA that you've banked, and that there are unclear ethics around it, and that there's concern that people are using the blood, especially if you don't use it, for research later. So it's like a lot of this stuff that, which just means they'll have to put in place a form that you sign that's saying, no, I do not consent to that blood being used. You must destroy it, which is very obvious to me, but just seems very shocking. But I think a lot of people are storing their umbilical cords, the cells in the blood, especially because there are things like childhood leukemias and things like that you can use that blood and those cells to help the child itself. So I think that's a huge area right now, is trying to store things like that, and people are taking advantage of it. But there are issues, as there always are. Yeah. Any other questions? This other thing that I have here will be given to you, but it's a paper on the challenges in the treatment, and that it's a long way from bench to bedside. So it's an actual paper that you have. I'll give you, included in this is a link to a PDF where you can read about where the current research is now. But just the idea that it's pretty far from actually happening in, what it means, of course, to say bench to bedside is the scientific research bench and then bedside to the actual patient. So like this man who was in that video, he's actively involved in trying to work with the scientists and be basically a test subject for a lot of these things, which a lot of people do. They're in trials to try to help move research forward. But, and as he said, there's an urgency for some treatment or cure, but there isn't one yet. And so he's donating his DNA and donating his time, but he right now isn't getting the benefit. There's nothing bedside right now. Did I see a question over here, Dan? Yes.
I was just going to ask about the use of stem cells from one person's umbilical cord being used by somebody else, and is that where the controversy is as well?
Yeah, so yeah, for sure. So using stem cells from one person's umbilical cord to another person, there's controversy about that too, yeah. So who is allowed to access that? And also how it would be like transplanting, just like any issue that we have with transplantation, rejection. So it will have the wrong DNA, and it would still have to be a very good match. And so it would probably be for a family member, but they'd have to be extremely good match. And as you might know, and you know, from different transplant stories of bone marrow transplants, kidneys, livers, hearts, all those things, it's very difficult to not get rejection. And so, but there is a lot of controversy around who owns that and all that sort of thing, yeah, and how effective it would be, yeah. Yes. Yeah, that's the problem, is that it's not very successful right now, and it's really expensive. And so that's why, you know, lots of people think, so we hear, and that's why I kind of wanted to show that video and provide you with this paper, that it's not as close. So this is a little bit of the problem, I think, with medical research. It's not really a problem, it's just the way the system is set up. But you hear that doctor and scientists talk, and they say, oh yes, this is, you know, really great. And if we can do this, you know, we take these cells, we put them back to stem cells, and then, you know, it sounds like, if I were watching the news and I heard that, I'd think, oh, we're using stem cells now to cure Parkinson's. That's great. And they market it that way, and you walk away thinking, well, we're just so close. This is amazing. But they do that largely because they want people to care, and they know people do, and they need money. And so they know they're on a right track, or they think they're on a right track, and so they want you to give money towards it. But the media is just infamous for making it seem as though we're like centimeters away from, you know, basically almost every day I hear something like, we're almost curing cancer. Like, curing cancer? Like, cancer's like so many different things. Like, which type of cancer? And like, that's just a huge statement, and I don't know how helpful it always is. So I think we're pretty far away, and it's very expensive, and it's very controversial, and the laws are not in place to protect people at every step yet. Did I see a question somewhere else before I go on? Okay. Okay, so do I hope? Oh yeah, of course. I do. Yeah, if we can take our own skin cells, if I could take my own skin cells when I start getting, if I know, oh, I have Parkinson's, or I start showing Parkinson's symptoms, and someone says, you can take your cells and just have them reverted back to stem cells, and or whatever, or they develop new drugs as a result, yeah, I hope so. Yeah, that would be great. Lots of people would not have to suffer. I think more than anything, what we aim for with lots of these things in the first steps is control of symptoms and control of progression and a better quality of life. It's not as much cure as that. And so I want to talk about depression and anxiety just before we move on to the new things today. And we briefly touched on this before. But I just wanted to talk a bit about how much the genetics plays a role. So 40% of depression is thought to have a genetic link. We watched a video, I think it was last time, that said about 50% for depression, or mental illness, I think. So it's pretty high. So if someone you're related to experiences depression, there's a really high chance that you will also. Parents or siblings with depression are 3 times more likely to have the condition than the average person might have in the population. So there's a very strong genetic component to depression. And so I'm just going to show this quick little video about that.
One question that frequently comes up is about clinical depression and genetics. This means if you have been diagnosed with this illness, how much of it is due to things like your lifestyle, chronic stress, other medical issues, and even substance abuse, and how much is due to the genes that you inherited from your parents. Researchers generally agree that if you have relatives who have suffered from a mental illness, the likelihood that you will or could be diagnosed with clinical depression is about 50%. While this does not mean that you may have to battle with this illness during your lifetime, you may very well be more at risk. This is like saying that someone who lives with chronic stress is more likely to experience the illness. As the risk may be greater that you will be diagnosed with clinical depression if your parents, grandparents, or other relatives experienced it, you may wish to take greater precautions to maintain good mental health. Of course, if you begin displaying any of the symptoms, you owe it to yourself to let your medical doctor know of your family history. Also, your doctor will most likely recommend certain lifestyle changes, such as surrounding yourself with positive friends, carefully controlling the quality of your entertainment, staying happy and laughing, watching your diet, and controlling your stress levels. However, if you are diagnosed, medical doctors know just what to do. They will most likely recommend a combination of psychotherapy, antidepressants, and even possibly transcranial magnetic stimulation should you not respond adequately to the other options.
I'm Dr. Glenn Archibald.
So this sort of goes into their center. So I just wanted to, what I'm trying to mostly get across, I guess, is that there is a huge genetic component to some of these things, maybe larger than you originally thought, but it just keeps coming up that it's also about diet, exercise, lifestyle, that you can alter these things. You don't have to just resign yourself to the fact that you will get this. There are things we can do to moderate and modify those genes and those risks, and how important those things are. Because, drugs are helpful and important and definitely work, and in combination with other changes as well. And so anxiety, similar, like depression, there's some genetic causes, but also environmental and lifestyle. Around 30 to 40% of the variability is related to genetic factors. So the variability in the type of anxiety that people experience. So there are many different types of anxiety and many different levels, and that is largely thought to be due to genetics. So how much you inherited from people in your family. So overall, from everything that I talked about there and last time, that alterations of genes can cause mutations, and that can lead to dysfunction in the body. So we know that. That's what is underlying these diseases and disorders. Variation in both elimination or resistance of certain factors can play a role in disease in organisms like us. So if we alter or change our environment in some way, we can change the disease course. Disease and disorders are preventable by supplementation or bringing the body back to homeostasis. So homeostasis just means not the disease state, like in homeo means same, stasis means same, so just back to normal state. And there are many diseases and disorders that have a genetic influence, but environmental and lifestyle choices are also implicated. So just as I was saying, it's not a sentence to have a mutation, and there are really important and useful things that you can do to try to alleviate some of that. And the last thing I just wanted to talk about before we get into the newer stuff was the genetics of obesity and overeating. And there was a lot of discussion and questions last time about that. How much is genetic and what do we know?
When we started this study, what we were interested in is trying to figure out what predisposes people to becoming obese. Certainly when you look at our current population, there are some people that are more obese than others, and there's a whole range of that. And we knew from previous studies that some of that was genetically predisposed. And to do this effectively, what we had to do is really bring together groups that could give us population-based data on people that have measures of obesity measured, and also that have been genotyped or assayed for different changes in their genetic structure, so that we can find genetic changes that correlate with people becoming more obese and developing metabolic disease. More than 100 groups around the world came together in a consortium called the Genetics of Anthropometric Traits Consortium. And we got a working group together to pull the data and then look to find specific regions of the genome that are associated with these conditions. And from that, as we put the data together, variants came out that were reproducibly associated with these traits, suggesting that they're real. And these variants were near genes that told us a little bit about the biological predisposition to obesity. We found 97 different regions of the genome that were associated with increased body mass index. And from these, we are able to sort of look near these areas and look at some of the genes that are in there and figure out what are the kinds of processes that may be related to predisposing people to obesity. Surprisingly, when we started off working on obesity, it's a metabolic condition, and so we were looking for metabolic processes, metabolic genes. But interestingly, one of the things we came across is that a lot of the genes, or some of the areas that we were landing in, actually played a role in sort of neuronal processes. And some of these neuronal processes that some of these genes play in make sense in retrospect. Some of them play a role in controlling appetite, but then some of them seem to play a role in energy expenditure, in physical activity. Some of them seem to play very basic roles in how neurons talk to each other or signal from one another. And so really that opens up a whole new area of understanding for what causes people to be predisposed to having higher obesity and higher rates of these other related metabolic diseases that we really weren't expecting. It was really very exciting to us as new ways to potentially understand this disease so we can do something about it. Obesity correlates with a lot of other metabolic diseases, such as higher rates of diabetes, higher rates of cardiovascular disease, higher rates of cholesterol problems, higher rates of blood pressure problems. And we also found variants that not only affected body mass index, but also affected these other traits. As people become obese, some people stay healthy, some people develop these metabolic diseases, and we don't really know why that is. And when they do develop these diseases, some people develop different sets of them. Some people are obese and have diabetes. Some people are obese and get heart disease. But now we are starting to find sort of genetic variants that predispose people to not only obesity, but some of these other metabolic diseases. So that's starting to get to a better understanding of, how this causes disease. You're defining the variants and then the genes that sort of underlie a predisposition to metabolic disease in general. So in the future, we may be able to subcategorize people into those that will sort of develop healthy obesity versus the ones that are going to get the other metabolic complications of obesity, again, so that we can better tailor their care in the future. One of the things that becomes very evident from our work is that there's a lot of different causes of obesity, and in particular that affect body mass index. We have 97 different regions of the genome that associate with this trait. So if people were waiting for one thing to come out of this that causes obesity so that we could cure it, that's not really what this condition is all about. We hope that we can look at you and sort of give you a better sense of, if you gain weight, you may develop obesity, but you won't develop any other diseases, or you'll become obese and be very at high risk of developing diabetes or cardiovascular disease, and sort of be able to subclassify people into what really caused you to get to this point and to have this diseases versus the person next to you.
Okay, so I just wanted to touch on some of those things in there. So a couple of years ago, I saw a talk here at UVic, and some of the physicians, it was some physicians from here, there was a collaboration of people doing a talk, and there's a new disorder now called diabesity. So people that they're trying to say they're people, and it's like she's saying that as they gain weight, they get diabetes. And so it's very serious. And she's talking about people who sometimes become obese and don't aren't more predisposed to diabetes or heart disease or any of these other things. And so it's interesting to the way she's talking, but mostly what she's saying, which I think are some good points, is number one, a lot of these things, we're not looking for one problem and a cure. It's just understanding information about, okay, so we know that obesity isn't good for us, and here's some evidence that actually not only are you gaining weight, but you're increasing your risk for heart disease, you're increasing your risk for diabetes, etc. And we know that too, but there are genes associated that you may be able to know, oh, I have 82 of those 97 genes. I better be careful. I better make sure that I eat well and exercise and don't allow that weight gain to happen if possible, because we know, as she's saying, it's a lot of genetics and behavior and appetite and things like that, but mostly just to gain information about the link between genetics and some of these things. So are there any questions about the obesity part of things? Okay, I just wanted to make sure that I touched on some of the questions that we had talked about last time. So I'll just talk about this for a couple of minutes, then we'll take a break. And this part here is pretty short, and so I wanted to talk about it just so that we could take a look at my 23andMe thing, if that's interesting to you. And so what I wanted to talk about now is why all of this genetics and everything I've talked about for the last 3 weeks and all this videos that you've seen, why any of this is really important. We've talked about it's important for you knowing your family history and what risk you might have of having some of these things. But we've also talked about what was the reason to do this Human Genome Project and this $17 million it cost, and what good was it? And I mentioned that I read an article that Craig Ventnor had written about how he said it largely told us nothing. It told us that there's lots of junk and we don't really know, and so what good has it been? And I think the big thing is that we just don't know yet. There's a lot of data and management of that data and sifting through that. But the hope is that we'll get to a place where we can have a personalized medicine so that you walk into a doctor's office or a hospital and your care and your treatment and your drugs are tailored to your genetics and you specifically, and how you metabolize drugs and how you deal with anesthetic. So has anybody here ever had general anesthetic before? Okay, so, and did they, I don't know how, sometimes anesthesiologists don't talk to you at all, and sometimes they're really friendly and they'll talk to you. And so has anybody ever said, so especially women are very sensitive to certain types of anesthetic, and they'll often warn you. So I remember getting a warning that they said, so you might wake up like really, really upset. And so like just crying and upset. And it didn't happen to me, but a woman, a couple over recovering, was really upset. And so women just have a different way that, in not all women, but there is, like, they've noticed that there's a difference in the way women metabolize and process certain anesthetics. And then also, I also have, I'm pretty much a redhead, but I'm not, like, I'm reddish head, but red hair. And people with red hair do not respond very well to certain anesthetics either. There's lots of interesting things about red hair people. I should do a whole thing on red hair. It's really kind of cool because there's lots of more prone to celiacs, higher pain tolerance, really interesting things. But also anesthetics. So we're not as sensitive to the anesthetics, so you have to be really careful because you need more anesthetic for people with red hair. And then there are different things about ethnic backgrounds, etc. So we already know some very basic genetic differences. But what the hope is that we'll get to a point where we can separate people into different groups and know, oh, okay, this is a person who's from Italy and who has this genetic makeup. We should use this set of drugs on them to prevent these side effects that they will get from this, etc. And so the hope is that these medical practices will be tailored to accommodate for the unique needs of every individual, or at least groups of individuals. And it's based on their predicted response or risk of disease. So they can predict, just like that, like I've had anesthesiologists say, you might experience this because we see this often in women, or you might, need more anesthetic because you have red hair, or like they have a prediction, and this is just helping. The more we know, the more personalized it can get. And this dates back as far as Hippocrates, where the professional was expected to treat the patient rather than the disease. And so now we're trying to get back to that in medicine, where it's not about, I'm going to treat your lung cancer. I'm going to treat you and your specific needs and you specific genetically in the context of having this lung cancer. But not everybody responds the same to all these treatments. And I don't know if by any chance a couple of weeks ago anyone happened to hear on CBC, there was a really good program about, there's a big push in NIH, the National Institute, I can't remember what it stands for. Institute of Health, yeah, thanks. I always want to say CIHR, but NIH. And so they have a real push, and there was a woman who's the chair of research there speaking about how all or very often research in mice that I've talked about a lot has been done on male mice. And all the drugs have been tested and everything has been tested on male mice. And so all the drugs that we have and all the treatments that we know, they don't know what it does in females because and there's a history of a very good reason why that happened is because, so it's a very good logic, but then it all falls apart when you really think about it, is that because of pregnancy and hormones, they didn't want to test it in females because that would confound things. But women get pregnant and also get sick, and sick people are pregnant. And so we need to know what happens in a pregnant mouse, and we need to know what happens in a female mouse. Also, female mice are way more expensive. So it costs a lot more for them to do the research on female mice. And so they're trying to cut their costs, and they do everything on male mice. And so so when, she's arguing, and lots of people are arguing, that when women go into a doctor's office, or into a pharmacist or anywhere, their treatment is based on what has been shown to work in men. And so it doesn't necessarily work in women. And it's been shown to, and it's also the same for men. This has been shown to work in, like young adolescent mice. Well, what does that mean for all of us as we get older? And men are not the same. And, so this is what all this personalized medicine is about. And the NIH push is to obviously increase that to females, pregnant female mice, etc., so that we know how to treat a pregnant female person if that were to happen, which of course it does. And so the Human Genome Project that I've mentioned quite a bit was this project to sequence the entire genome. And it was this international research effort to sequence everything, and it was completed, which is shocking, 13 years ago, but in April of 2003. And so it allows us to hopefully at some point get to the point where we understand the blueprint for what makes a human. And personalized medicine can use that data to highlight trends that happen throughout the species. So we can see that, most women have this version of the gene, and that doesn't work very well with this drug, so maybe we shouldn't give that drug to women, or etc., so that we can look for trends in women versus men, different age groups, different ethnic backgrounds, etc. And diagnosis and treatment of genetic diseases can occur more quickly and efficiently because we have the information now. So you can know for sure if you have the mutation for Huntington's because we know where the mutation happens in Huntington's. So all this information is good, largely because it increases the likelihood that you'll find it and you'll know in a timely fashion. Okay, so I'll wait until after the break to show the video, but are there any questions about personalized medicine or anything to do with mice or anything like that before we take a break? Sure. Mm-hmm. Yeah, so there is such a thing as seasonal affective disorder, so where people are affected by seasons. But I think that's just sort of a universal, even though it's not always true, but way that they represent depression is this feeling of being down and low and like you're being rained on and like, but it's not always true, but I think that's what they meant, was that that's the sort of stereotypical way that we think about depression. But it's true that there is such a thing as seasonal affective disorder, and people in, Vancouver, never Victoria because it's so beautiful, but Vancouver and Seattle, And of course here it's true. Anywhere it can happen. It's just like if you don't get enough sunlight, you can get those light boxes that help and give you some light and some relief from it. Seasonal affective disorder? Probably is. There's probably some genetic component to it. Yeah. Just like I guess the kind of the point of almost all of what I'm saying is that there's almost a genetic component to everything. Every single thing, every characteristic we have, every behavior, everything, but that's not all there is. In something like seasonal affective disorder, and even lots of depression, anxiety, it's more environmental and what you do. So there's a genetic component, but even more so, it's other factors that you might be able to alter. Okay, so, sorry, David.
Yeah, just one kind of last question, I guess, before the break. I kind of listened to a radio program out of the States, and they keep advertising. It's a public broadcasting station, but they say there's, I think, a Swedish clinic, and they deal more with cancers. And they are claiming that their treatment is based upon your genetic makeup, so it's individualized. So the question is, how close are we in certain diseases to actually being able to take the person's genetic makeup and then actually tailor treatment to them?
Yeah, so that's excellent. So for anybody who might not have heard at the back, so there's on American radio programs, they advertise this clinic where they claim that the treatment for cancer is based on your personal genetic profile. So it's tailored to you. And how close are we to that? And someone brought up a couple of weeks ago this immune-based, like immunology-based treatments for cancer. Why can't we stimulate our own immune system to kill the cancer or to not attack ourselves in autoimmune disorders? And that's where all the research is right now. So even the local BC Cancer Agency here, they're very focused on immune therapy, and that's largely based on your genetic immune profile. So they can look at what's called your human HLA, your human leukocytic antigens. So that's just these proteins on the outside of all of your cells. It's what they look for to try to match in transplants. They can profile you and know what your HLA profile is and then tailor a treatment towards that. So there are some things happening, and there are lots of trials in cancers. There's an excellent trial going on right now. Well, hepatitis C is basically curable now. So they're almost at the stage of curing hepatitis C, and it's largely based on personalized knowing your immune system. And there's a huge trial going on right now for kidney cancer, and it's largely based on your own profile of your immune system and working with that in order to tailor treatment to it. So there are big steps and things happening. It's just there's a good and a bad thing. Canada's really extremely conservative about medical intervention and drugs, which has a good and a bad side. It's bad if you're waiting for it because it'll take forever. So a good example is fecal transplants. So for ulcerative colitis and things like that, we know that if you transplant the bacteria from someone who has good flora of bacteria into someone who doesn't, you can almost cure these things. But because people hear the word fecal, you're not actually taking fecal matter. You're taking just you could take a pill, but there's lots of controversy around it. It will take years before that will ever actually end up in stores. It's illegal right now to do it. There's a guy in Calgary who wrote a textbook about how to give yourself a fecal transplant because he just thinks it's so incredibly important and people should be doing it, but he's not allowed. And so what I'm trying to say there is that we know some of these things work, but they're not out there yet because there's so many legal things to go through before it's out there, because we have to be very careful that we don't harm people in the process. Okay, so let's take 15 minutes until 20 after, and then we'll come back. Maybe we should get started. Okay, so I'm just going to get started with a video on how to sequence the human genome. And then I had a question that I'll talk about in a moment about how did we go from $17 million to sequence a genome to now you can do it for a couple of $100. Like how did that happen? How did it cost so much money to do it before and now you can buy it for sometimes $99? So I'll show this video first and then we'll talk about that.
You've probably heard of the human genome, the huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is a genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA. And DNA is made up of long paired strands of A's, T's, C's, and G's. Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion.
Oh, wow, $3 billion.
But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome all in a matter of minutes, and for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big and very, very small. The individual letters of DNA, the A's, T's, G's, and C's, are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn. So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces. Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the ATGC sequence of 2 pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we have to read them all somehow. To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on. So the next step is to decipher what the sequence means. how your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.
Okay, so I'm not sure how helpful that was to try to teach how the genome is sequenced. It's still this kind of mysterious, like, well, they just break it up into millions of pieces and add these colored letters and then you read them. So it's obviously, hopefully maybe obvious, but all just with computers. So it's all just done where what they're saying with those colored letters is they tag the DNA with these colors that bind. So the colored A will always bind to a T. G will always bind to a C, etc. So then they run it through a computer like a laser that picks up the colors and tells you the order of those colors, and you just translate them backwards. Then it'll tell you what the other strand was. And so now, back to the question of why did it cost so much money to do this in the first place, and why did it take so long, and how is it possible that I can send off my DNA and get this done so cheap now? It's largely because if you think, so this video was done in 2013, and so we're talking about it being done 10 years ago, and it took 20 years. So now we're almost 40 years from when they started. Things have changed dramatically in terms of computers and speed and processing, lasers, all that stuff. And so that's all cheaper. People know how to do it, and it's faster. So if you go back to that video that I showed in the very first week and then the second week again about how it would take someone 50 years to type out the entire genome if you're typing 10 words a minute for 8 hours a day. That is like that, of course, that isn't happening anymore. The computers are way faster than that, and so they can do it faster and much, much cheaper. So that's one answer. The second part is that when you send your DNA off, so when I show you my 23andMe, they're only testing certain parts of the genome. So the stuff that they think right now is junk, they just completely eliminate, and they don't even look at it. So remember how much that was? Like 98% is junk. And so then that rules out a whole bunch of work and a whole bunch of time. So just to sequence the part that's important is significantly less. So that's partly why it's so much cheaper and so much faster now, because they've eliminated that information. So are there any questions about the sequencing? Yes.
How do they determine the beginning and the end?
Yeah, the beginning and the end. It's a good question. So, so as you might remember that I showed you that all of like our chromosomes, so when we take a look at our chromosomes, in the karyotype, they look like this. Remember, there's like 2 chromosome 1s, these are 2 chromosome 2s, etc., all the way to 22, and then we have X and Y. So we know that there's a beginning and the end to this chromosome because it's just a finite length. It's not round, right? Like, so it's one length. And then we also know, so remember the telomeres? So telomeres are the caps at the end. So our DNA is like this big long strand, and then we have at the ends of our chromosomes, we have these caps on both ends. These are the things, remember, that tell you, which I also found, I was going to show you this video, but I found this place in the States that will sequence the length of your telomeres and tell you if they think that you're older or younger for your chronological age. But of course, it costs a lot of money. And so these telomeres at the end are there to protect the genes. So then the genes, like this might be a gene, and this might be a gene. And what's kind of cool about replication of DNA, or interesting anyway, to me maybe, but is that the machinery comes along and starts replicating the DNA, and it always starts here instead of here. It's just the nature. So you're always going to, every single time your cells, that's why it's a measure of aging. Every time your cells replicate and the DNA replicates, you lose a little tiny piece at the very end. And then next time it does it, you lose a little bit more and a little bit more. And the telomeres are there so that it's just telomeres you're losing. They're not important. They don't have genes in them. It's protecting the rest of your DNA. So that's why it's a measure of aging. How many times have those cells turned over? And some people are rapid agers and their telomeres shorten really fast, and some people are slow agers and it turns over really slow. And so this cap, so what this is this repeat, TTAGG, repeated, that's what the little N means. It's like that repeat is repeated over and over and over and over again in a sequence, and that's your telomere. So TTAGG, TTAGG, et cetera, et cetera. And if you have, let's say, 100 of those, then this is arbitrary, but that might be a very long telomere. And then if you had only 50 of these repeats, now it's a little shorter. And let's say you only had 10, that's very, very short. So and then they can look at the TTAGGG and see what came just after that. And that's where the genome actually starts. So they have these caps on the ends of all of the chromosomes so they would know where it begins and ends. Yeah. But it's a really good question. They have to single out the chromosomes. They don't put all the random DNA on there together. They would have no clue. Yeah, like how do you even know how these pieces fit together? Yeah, it's very complicated. Was there a question? Yes.
So even though they don't have much use, these people that live to be 115 years
Yeah.
like that, is maybe the little caps aren't wearing?
Definitely.
And the babies that
Sure.
die?
Sure.
And are aging before their time?
Yep. Yeah. Okay. So the question was, even though these telomeres don't really have a function, could that be why people live to be like 113? Could it be that these telomeres are just not shortening at the same rate? They're staying longer. And the answer is yes. So we know that there's a link between the length of these telomeres. So these people that live to like 113 maybe started with like 1,000 repeats, where the average person starts out with 500. So they're already starting off their life with super long telomeres, and then maybe the turnover is just slower. And so and they shorten less. And also, just to complicate this, but people asked about this in the first week, there's this enzyme called telomerase that adds length to telomeres. So its job is to add some length to these telomeres. And some people have a lot of telomerase, and some people don't have a lot of telomerase. And so that might mean that their telomeres never really shorten. Maybe the people that live to 113 have tons of telomerase, so every time it shortens, it just re-lengthens and they stay really long. And then the second part of the question was young children that die or age really rapidly. Is that what's happening in them? So it can be, but one of the most common examples is progeria, that genetic disease progeria, where they age super fast, like where they're 2 years old and they're functioning like a 50-year-old, and very, very quickly. It's way more than that. It's not just the telomeres. There are other genetic problems, but they do have extremely short telomeres. And so just going back to what we talked about with Dolly, the sheep, she was born from an adult cell, and so she was born with adult length telomeres. And then as she aged, they shortened, and so she was 2 and had 12-year-old sheep length telomeres. And so she aged very rapidly, and she got arthritis and all these aging related diseases because her telomeres were really short. And then I think I might have mentioned, so for the benefit of any people that weren't here, that we've looked at IVF and other like ICSI, so IVF is in vitro fertilization, so test tube babies, and ICSI is intracytoplasmic sperm injection, so injecting the sperm right into the egg in a dish. Children born from these, in particular ICSI, I studied this when I was in school, they have extremely long telomeres. So way beyond reasonable, really long. And we don't know why. And obviously we've manipulated something and that's happened, but is that a good thing? Does that mean they'll live really long? Or we also know that sometimes when telomeres get too long, they can get tangled up and cause other problems. And people might be more susceptible to things like cancers. We definitely know that as the telomeres get really short, there's susceptibility to cancer. So, but we don't know what the consequence of really long is. Yes?
I wanted to get back to an umbilical cord question.
Oh, sure.
That's similar to IVF. So when an embryo has been fertilized by the mother and the father, but is implanted in a surrogate
Yes.
mother, where the nutrition is coming through
Yeah.
an umbilical cord, do they share DNA, or
Yes.
does that affect telomeres?
Yeah, excellent question. So this is another whole other really fascinating thing, is that, yes, so the question was, if there's an IVF, so an in vitro fertilized test tube zygote implanted into another person, so into a surrogate, do they share DNA? Because they're sharing nutrition. Do they share DNA? And so the surrogate doesn't, that zygote is genetically the original sperm and egg. But through the umbilical cord, it's more in the direction of the mother. So from the baby to the mother. So she's constantly being infused with the baby's blood too, and receiving the baby's blood. And so this to me is just absolutely fascinating. Any woman who's ever been pregnant, whether you knew it or not, so that's another thing, is that lots of women don't even know that they were pregnant at some point, have forever in their body the cells and the DNA of those babies, no matter what. And so what's really fascinating about that is that we know that lots of autoimmune conditions like arthritis, so rheumatoid arthritis, scleroderma, lots of these conditions that are autoimmune, if you look at the joint or the skin or where the scarring or the tissue is, if you take DNA from that, you'll find baby's DNA in it. So it's like what your body's doing is your immune system is reacting to and trying to get rid of this invading DNA, because it's come through the umbilical cord into you. And so it's less that the surrogate is giving any genetics to the baby, it's the baby's giving genetics to her. But all that stuff about epigenetics that we talked about, what she eats, how she exercises, if she's around cigarettes, that's all impacting the genetics of that baby that she's carrying, way more than we used to think. Like, of course we knew if you had a surrogate, you would want them to be healthy. But now we understand it's much more serious than we thought.
Along those same lines, then, again, listening to CBC and being a mother of a son, they said that they have found the male gene cells in the mother's brain.
Yep.
So they were just speaking about males. But
because males are so much easier to find because there's a Y chromosome. Yeah, so it's just really easy. Like if you look in females and you find Y chromosome material, that's not yours. Yeah. So it must be your child's. But now we can find the females too. Oh, but yeah, so we used to only be able to find the males, and it's much easier to find the males. But yeah, it gets in the brain, it gets everywhere. And so it has huge impact, of course. How couldn't it? Like there are cells and DNA sitting in your brain and sitting in parts of your body from a pregnancy. Yeah, it's fascinating to me. And that might be a trigger for a lot of these autoimmune conditions. So you can imagine your body's like, what is going on? Why do we have this male DNA like in our body? It's one thing to have the male fetus in you. There's already problems there, with compatibility, but then the cells are now in your body. It's fascinating to me. I just think it's amazing. Yep. Yeah, I think you asked that before. So why can't we just take telomerase and increase the length of our telomeres? Because we can't get it into every single cell. Like if I took telomerase, it would get into my digestive system, it would get into my bloodstream, that's it. Like it's not going to get into my brain cells, it's not going to get into my liver cells. Why? No. Oh, but that's because lactase is not acting in your cells; it's acting in your gut. And so what happens with so it's a really good question and example. So why we take enzymes for digestion all the time? We can take lactase, we can take things like that, but it's not happening inside the cell. It's happening in the digestion. So telomerase getting into a cell, it would have to get in like all billions of cells that we have, right? So and it wouldn't really help. Greg, are you trying to help me here? Yeah. Thanks. Yeah.
Recent exercise studies are showing that exercise influences telomerase activity.
Yep.
Specifically tied to androgen
Oh, interesting. Yeah.
And shows an effect about 24 hours after a single bout of exercise.
Yeah, thanks. Yeah, and that's just is all related to, so the epigenetics of changing the genes, and then it lengthens the telomeres. So it's telomerase activity, and they know that, like as Greg just said, but it also lengthens the telomeres, just exercising. Like we know that for sure. Yes.
Just in relation to that comment, I understand from what I've read is that intense weight training, strength training, also has the same effect as well as aerobic exercise. But strength training, again, it impacts the system to the extent that it does help lengthening the telomeres.
Yeah, everything. I know, it's amazing. Like exercise is amazing. Yeah. Did I see another question here? Yep. Yeah.
Do this telomerase stop lengthening? I mean, is there a time when exercise will not influence that, or?
It's a good question. Yeah, is there a time at which the telomeres will just stop lengthening? Is there an age at which, no matter how much exercise you do, that won't help? I don't think we know that, and I doubt it. I would think that it's just going to continue to, maybe the effect gets a little less or something, but I don't think that there's an age at which it would stop. Like, I think that if anything, it's always going to be good. Yeah.
Thank you.
Yeah, you're welcome. Okay, so I just can't remember what else I had. Oh, the pharmacogenomics and pharmacogenetics. This is like 1 or 2 slides here that I wanted to show you. And then we're running out of time, but I kind of did want to show you my 23andMe, but we don't have to. Okay, but just pharmacogenetics is just looking at, I already touched on this, but what inherited differences do we have, and we all do, that cause differences in how the drugs are metabolized, and what pathways are used in our body, and how do we respond to those drugs? And like we talked about, and I mentioned, and you probably know this just from yourself, that everyone's different. Some people, like I have zero reaction to caffeine. People that say like, oh, I have to have a coffee in the morning. I'm like, I don't even understand that. Or I'll drink black tea at night and someone will say, don't do that. You'll never sleep. I'm like, I don't even know what you're talking about. I don't have that. Like, I just don't respond. And some people, they're just through the roof with caffeine. And so we know just anecdotally that things like that affect people. And that some people take, an Advil and their headache goes away and some people it doesn't. We metabolize things differently. And so this is taking a combination of the pharmacology, which is the science of drugs, and genomics, which is everything we've talked about, the study of genes and their function, and putting them together. So if your genes function in a certain way, to metabolize things in a certain way, then those drugs will work differently in you. And so the therapeutic effect is the consequence of medical treatment where the results are thought to be desirable. So that's what is meant by therapeutic effect. So what's the therapeutic effect of this drug? How does it work, and how is it beneficial? And an adverse effect is when something's harmful or undesirable, or is also called a side effect. So what we're looking for with pharmacogenomics is to increase that therapeutic effect and decrease the adverse effects. And it's the same with side effects. I'm sure you've all experienced where some people can't take certain things. They just, I just always get this side effect, and you've never experienced that. And it's totally fine for you. And as we all know about horrible things that happen, people die from taking some drugs because of side effects that the rest, like very small percentage of people will just die from taking this. And it's the same with illicit drugs. We hear about people dying from taking, I don't know, like maybe ecstasy or something like that. So you hear about, I don't know, it's not always just teenagers that are taking it, but you hear about young people trying a drug for the very first time at a party and they die. Where somebody else takes a drug for 20 years and it never affects them at all in any bad way. And so we're all different in the way we metabolize things. So that's what pharmacogenomics is trying to do, is work with that and make things better for us.
The right medication can save a life, but a medication that works well for other people might not work for you and may result in significant side effects. Pharmacogenomics is the study of how our genes affect the way our bodies respond to medications. Because of their genetic makeup, some people's bodies break down certain medications too slowly. The medication builds up in the body, causing severe side effects. Other people's bodies break down the same medication too quickly, eliminating it before it has a chance to work at effective levels. Yet others may be predisposed to have significant, sometimes life-threatening side effects from a medication simply from having the wrong genes. Scientists at Mayo Clinic are investigating how specific variations in genes affect how well certain medications work in the body. Using this information, doctors can examine your genetic profile to predict whether a medication is likely to help you or hurt you before you even take it. That's the promise of pharmacogenomics, providing the right drug for the right patient at the right dose. For example, if you have high cholesterol, your doctor might prescribe one of many statin medications to lower your risk of a heart attack. But these drugs also can have significant side effects. Finding that out can be expensive, time-consuming, and could delay receiving proper treatment. Pharmacogenomics offers a better way. A genetic test can show how your body is likely to respond to a certain statin. That helps your doctor prescribe the right dose of that medication, or even a different medication to get the best results for you. Today, a variety of genetic tests are available to patients at Mayo Clinic. One genetic test already available can help predict how an individual will respond to more than 30 types of medications. These medications are used in the treatment of heart disease, breast cancer, and psychiatric disorders. Researchers at Mayo Clinic are advancing this new science, identifying more genetic variations that affect the body's response to specific medications. Knowledge that can transform the practice of healthcare by incorporating this information into routine clinical practice. Find out how you can help Mayo Clinic bring the...
Well, Mayo Clinic doesn't help us very much. Of course it does. Their research does, but we can't go there unless you want to pay a lot of money. But as they're talking about, and as I've said, this is the promise of pharmacogenomics. We're not, like, there are some things that Mayo Clinic can test you for, and why some private clinics can test you for certain things, but we are not that close here. And we're not that close to actually sequencing our entire genome to be able to really effectively know which drugs to take. But we're getting there, and people are doing a lot of research on it. It's a huge field of study to try to figure this out, because it is very, very important. Okay, so just a little summary here. So there's a large variation seen in the genetic differences and predispositions that individuals have, as well as lifestyle and environmental, as I keep saying. Personalized medicine treats the patient rather than the disease characteristics alone. That's the hope, is that you as an individual are very different than you as every other person who has diabetes or every other person who has liver cancer or whatever it might be. You're unique and different, and how can we use that in medicine? Trends studied in the past, including the Human Genome Project, can help us correlate to set point when helping an individual. So what exactly are the genes that you carry? What are we trying to aim for? How does your genetics work with the treatment? And then pharmacogenetics or pharmacogenomics is looking at the drugs that we can use specifically for certain individuals and be able to predict the outcome better of the therapeutic and the effects and the versus the adverse effects and how effective the treatment will be. So that's the hope. That's what all the personalized, wow, I actually timed it fairly decent, as long as you don't want to, if people, so that's all I was going to say. And unless there are any questions, and if I'm really happy to, I would actually think it's really interesting, and I was just looking at it the other day. I would be happy, but if anybody has to leave, I understand. But I'll show you my 23andMe and just how interesting that looks. But are there any questions about anything that I just talked about at the end there? Or anything at all, really? Yes?
Is it likely that food sensitivities would react the same way as drug sensitivity?
Yeah, so the question is it likely that food sensitivity would react the same as drug sensitivity? And yes, for sure. So we know that there are lots of genetic reasons why people have certain food sensitivities. So some really common and basic ones are things like celiacs, the sensitivity to gluten, inability to, so like pernicious anemia and inability to absorb B12, things like that are all genetically based. And so if and lactose intolerance, things like that, enzymes that are missing is all genetically based. And so food sensitivity for sure would be very, very useful to know, especially because we're talking about how important diet is, but it's not standard. It's not like you can follow the same diet for every person. It's very individual, depending on any underlying conditions and your genetics. So I can't eat gluten, and that's another redheaded thing. I think I said that, celiac, yeah. I can't eat dairy, and so telling me to eat lots of fiber and grains and dairy would be harmful to me. And people with celiacs that have a consistent inflammation in their digestive system are more prone to cancer. So it's very, very important, food sensitivities and your genetics. Yeah. And yes. Mm-hmm. I would predict that within 50 years we will be we'll be much better. I think the big thing is going to be we'll be much better at maybe kind of understanding and appreciating the impact of diet, food, and exercise and environment on genes. And then I think that we'll be much further along with our study and information that we have from the human genome and drug studies. I think that it will be closer to personalized. So things like that happening right now at the Mayo Clinic is really promising. You can go into the Mayo Clinic if you lived in the States. You could go there if you wanted to, if you had money from here, and get personalized. They would test your DNA and say, we're going to give you this drug and not this one because you have this gene that would make that drug, you'd have bad side effects. So that's really promising, and that's happening right now. And so I think that will be pretty common, that you'd be able to go in, have a routine little test done of your DNA, or maybe the doctor would say, get this done before you come, and then we'll look at everything. The problem is going to be, who are these doctors, and how do they know how to interpret all this genetic information? So, if my father's a doctor, and when he'll ask me things like, when growing up, my younger half-brother was in school with a child with Fragile X, and he said, can you tell me what Fragile X is? What's the genetics of it? And so I started explaining it, and he said, oh, forget it, it's way over my head. I'm like, you're a doctor, but only geneticists know about this stuff. Why would he? Well, he's a GP, and it was like he went to school like a long time ago. So you can't keep up with everything, and so that's going to be a real barrier, is who are the physicians that you'll be able to go into and talk to about your personalized genome that will even know. If I go in and talk to people with the information I know, talk to physicians, they just stare at me blankly like, I don't know, sure, I guess. Like they don't know. So I think that we're going to have to increase training for physicians, but I think we're really close. I think in 50 years it'll be much, much more personalized. Any other questions before I...
Your conference is scheduled to end in 2 minutes.
Forget about that. So that will stop recording in 2 minutes. Anything else before I show you? And obviously anybody can leave if you want to. Okay, I'll show it to you. Again, you'll see that some of the things are really fun and interesting, but some things are not good for insurance. Yeah, I'm denied insurance because I had cancer. I had melanoma, I had malignant melanoma, so they're like, no. Based on this? No, not based on this, but because I've already been turned down, I'll never get insurance. I'll get basic things. But see, $249, you can get it. It goes down sometimes to like $99. Yeah, they have sales around Christmas time. It's like, yeah. Yeah, to buy someone a gift. Okay, so this is me. This is my overview. So they give you lots of really interesting things. So 80.1% British and Irish. Shocking. And so, yeah. But it's interesting. So if you go, so the percentage of your DNA that comes from populations worldwide. So then you can click on it and it'll show you. So I'm 80.1% British and Irish, 2.8% Scandinavian, 0.6% French and German, 15.7% broadly Northern Western European, and 0.8% broadly European. So that's where obviously I think it was pretty obvious to me that I was going to cluster in that area. But it's kind of interesting to see
Your conference is now over. Goodbye.
Okay, I can stop worrying about that then. Okay, so so that makes up me. So all those different parts.